Okay, so hello everyone, let's get started. So this class, we're going to learn some new concepts regarding the ecosystems, risk assessment, and if we have extra time, we can talk a little bit more about the hydrology. Okay, so the corresponding chapters of the book is chapter 5, 6, and 7. So I think chapter 5 is about the ecosystem, 6 is about the risk assessment. So these readings are actually optional. And hydrology is the chapter 7. Okay, so I'll also upload these book chapters for your off-class reading. Okay, so again, we can do a quick recap of our last class. So last class, we derived some differential equations, right, to talk about the performance of the flow reactors. Again, we basically started with the mass balance equation, right? The mass balance equation is or can be represented with a few abbreviated terms, right? You have the accumulation equals in minus out minus reaction. Okay, so we said that for the flow reactors, actually, we expect that the accumulation term to be 0 because normally these systems or these pollutant treatment systems, they're operating under steady state, right? So then you're only going to have in minus out equals reaction. And we said that for a batch reactor, we have the outlet concentration to be equal. Actually, here for the batch reactor, actually, the final concentration is going to be equal to the initial concentration multiplied by an exponential term, right? For batch reactor, we said that the challenging part or the disadvantage is that everything happens in batches, right? While in normal operation of the pollutant treatment, we want a constant flow going through the system, right? That's why we refer to the CSTR, which is a continuously stirred tank reactor, right? It's well mixed. And we derive that the outlet concentration is going to be linearly correlated with the inlet concentration. That's C out equals C in divided by 1 plus K theta. And the theta here is what we call the characteristic time, retention time, or residence time. And similarly, we can derive the outlet concentration for a PFR, the plug flow reactor. Right, we have C out equals C in multiplied by exponential minus K theta. Right, so we derive these two equations. One is with the assumption that, or with the additional assumption that the outlet concentration equals concentration within the reactor, right? And for the PFR, we assume that there's no mixing along the flow direction. Okay, so further, we did a comparison between the PFR and CSTR, right? We give an example. So basically, these are the equations. We're not going to go over that in this class anymore. So you can try to understand it more. And if you have any questions in deriving these, just let me know. All right. So we ended the class with a comparison between these two reactors, right? So by plugging the values, we found that the efficiency for the PFR is actually larger than the efficiency for the CSTR, given that theta or the residence time is the same, right? So also ask the question why the PFR is more efficient than the CSTR. That's because the concentration in the PFR gradually decrease, right? If the concentration is gradually decreasing, then the PFR will have a higher reaction rate, right? In this way, you have a better efficiency, okay? So then one thing we didn't discuss is, well, if the PFR is so efficient, why don't we build the PFR everywhere, right? Why can't we, let's say for the water treatment, why do we still have those sedimentation or those primary clarifiers? Why can't we just use tubes everywhere? Okay, so one major issue is that for the PFR, if you think about its geometry, it's a tube, right? It's like a plug. Okay, so for these tubes, there is, for one thing, in order to push all, or there's a large amount of water going through, you're going to require a high pressure drop, right? It's going to lead to a high pressure drop, and then you're going to need a very strong pump, and that's associated with the power consumption or the cost for manufacturing or for managing these systems. And on the other hand, if you think about its geometry, let's say if you have a tube, really made a tube that has a total volume of 5,000 cubic meter, right? At the same time, it has a diameter of 1 meter. Okay, 1 meter is already pretty large, right? The diameter is already pretty wide. So if the total volume is 5,000 meter cubed, right, we can calculate what is the length of this plug flow reactor. You can see the surface area is what? Is pi d squared over 4, right? So this is close to 1 meter cubed, or actually smaller than 1 meter squared, right? Let's just treat it as 1 meter squared. Then it means that you need to build a reactor that has a length of around 5,000 meter cubed divided by 1 meter squared. That's 5,000 meters. Okay, basically means that this tube have to be 5 kilometers long, right? This is not realistic at all, right? So because of that, we cannot build PFR everywhere, right? We still need this CSTR to achieve a lot of these pollutant removal processes. And the CSTR also does not require a large pressure drop, right? So the maintenance is also going to be more manageable. Okay, so this is further about the choice of the PFR and the CSTR. Okay, so in this class, we're going to learn some new topics about the ecosystems, right? And then talk about the population, or talk about the basically the risk analysis. So these are just going to be some general definitions. It's not going to involve much calculations in here, right? So I think the contents of this class is also going to be quite relaxing. So here, what I'm showing is a painting, right? So for those of you who really like these different styles of painting, you can tell that this is actually the Water Lilies by Monet, right? So actually, this person drew so many of these water lilies that I think almost everywhere in the museums or art museums, you're going to see a few of these paintings by him, right? So actually, the water lilies are always depicting a ecosystem, right? For example, you can have these green plants over here, these tall grass over here, right? You have the water lily, which are the main subjects of the painting, right? You can have fish below the pond or in the pond, right? Bacteria, you can have these trees here, right? So basically, the ecosystem or ecosystem is defined as a living system that include plants, animals, or microorganisms, and they can include many of the places that we normally deal with. For example, lakes, rivers, forests, lands, constructed wetlands, and so on, okay? And including the animals, right? The animals there can be so many types of them. Some of them are fed on grass, some of them are fed on, let's say, the other types of animals, right? So whenever we talk about these carnivores, for example, or the animals that have to live based on eating other animals' meat, right? So we have some negative feelings about them, because mainly we feel that, well, it's not healthy for the environment, right? And sometimes these carnivores can be very dangerous too, right? But actually, they're a very important section or important element of our ecosystem. Okay, for example, the Yellowstone National Park actually reintroduced wolves to help revise or help retain the or help to upgrade, let's say, the ecosystem that's happening in the natural system. Okay, so how does that work? So here I have a short video that I can share with you. Let me just quickly share that.
One of the most exciting scientific findings of the past half-century has been the discovery of widespread trophic cascades. A trophic cascade is an ecological process which starts at the top of the food chain and tumbles all the way down to the bottom. And the classic example is what happened in the Yellowstone National Park in the United States when wolves were reintroduced in 1995. Now, we all know that wolves kill various species of animals, but perhaps we're slightly less aware that they give life to many others. Before the wolves turned up, they'd been absent for 70 years. That the numbers of deer, because there was nothing to hunt them, had built up and built up in the Yellowstone Park, and despite efforts by humans to control them, they'd managed to reduce much of the vegetation there to almost nothing. They'd just grazed it away. But as soon as the wolves arrived, even though they were few in number, they started to have the most remarkable effects. First, of course, they killed some of the deer, but that wasn't the major thing. Much more significantly, they radically changed the behavior of the deer. The deer started avoiding certain parts of the park, the places where they could be trapped most easily, particularly the valleys and the gorges. And immediately those places started to regenerate. In some areas, the height of the trees quintupled in just 6 years. Bare valley sides quickly became forests of aspen and willow and cottonwood. And as soon as that happened, the birds started moving in. The number of songbirds, of migratory birds, started to increase greatly. The number of beavers started to increase because beavers like to eat the trees. And beavers, like wolves, are ecosystem engineers. They create niches for other species. And the dams they built in the rivers provided habitats for otters and muskrats and ducks and fish and reptiles and amphibians. The wolves killed coyotes, and as a result of that, the number of rabbits and mice began to rise, which meant more hawks, more weasels, more foxes, more badgers. Ravens and bald eagles came down to feed on the carrion that the wolves had left. Bears fed on it too, and their population began to rise as well, partly also because there were more berries growing on the regenerating shrubs. And the bears reinforced the impact of the wolves by killing some of the calves of the deer. But here's where it gets really interesting. The wolves changed the behavior of the rivers. They began to meander less. There was less erosion. The channels narrowed, more pools formed, more riffle sections, all of which were great for wildlife habitats. The rivers changed in response to the wolves. And the reason was that the regenerating forests stabilized the banks so that they collapsed less often, so that the rivers became more fixed in their course. Similarly, by driving the deer out of some places and the vegetation recovering on the valley sides, there was less soil erosion because the vegetation stabilized that as well. So the wolves, small in number, transformed not just the ecosystem of the Yellowstone National Park, this huge area of land, but also its physical geography.
Okay, so I think this is a very nice video talking about how the different elements of an ecosystem can play an important role, right? So specific for this case. So by reintroducing the wolves into the Yellowstone National Park, right? So we could basically make improvements to all the other elements of the ecosystem, right? So even to the geology. All right. So I think this is a short video, right? So there are some definitions associated with the ecosystems, right? So for an ecosystem, we have to understand their energy and mass flow, right? So specifically on this topic, right? So all the elements are categorized based on how they're generating the energy, right? So for example, or based on where their energy is coming from. All right, so there is phototrophs, right? Phototrophs are basically utilizing the sunlight as the primary energy source. For example, the trees, the grass, right? The phytoplanktons, right? The bacteria that's using sunlight to populate themselves, right? There are also chemotrophs. So chemotrophs are utilizing the chemical energy, okay? For example, the herbivores, right? So the animals that, let's say the rabbit or the deer, right? So they consume the plants, basically consume the phototrophs, right? To generate the energy. Okay, and these animals are generally characterized with little food, right? So for basically to avoid being captured by the carnivores, right? So on the carnivores, right? So basically those are the ones that are fed based on the herbivores, like the wolves or tiger or human, right? So these are also the secondary consumers, right, in the ecosystem. And generally, the carnivores are characterized with the sharp tooth, okay? And furthermore, for the phototrophs, herbivores, and carnivores, they can generate the energy through different types of activities, right? So for example, there's aerobic respiration. So this is a process that consumes oxygen to convert organic processes, organic components into energy and carbon dioxide and water, right? And there's also anoxic respiration. So anoxic respiration is a process that's used nitrogen as the source. And specifically, these nitrogen are in the state of nitrate or nitrite, okay, as their source, or as their oxygen to basically convert organics into the energy. And furthermore, there's also anaerobic respiration. Anaerobic respiration is a process that does not or cannot let oxygen or nitrogen present. For example, when we try to make yogurt, right, we add these bacteria into the milk. We cannot let oxygen to get in. Otherwise, the yogurt will just go bad, right? Or the milk will just go bad and we cannot get yogurt anymore. So basically for this yogurt making process or the fermentation process, we cannot let oxygen or nitrate to present, okay? Furthermore, there's the definition of the bioconcentration. Bioconcentration is a process where certain species, certain chemicals will get furthermore concentrated as they climb up the food pyramid, okay? So again, for the food pyramid, right? So this is basically categorizing all the elements or the living species based on how they generate the energy, right? So we have the phototrophs that's at the bottom of the food pyramid, right? Above these phototrophs, these are the herbivores, which basically feed upon these green plants, right? And further above the herbivores, there are carnivores, right? So they're relying on the meat that's coming out of the herbivores, right? And the bioconcentration means that some chemicals or some chemical species and their concentration can get higher and higher as they go up the food pyramid. Okay, so one example of the bioconcentration is the polychlorinated biphenyls. I think some of you also studied this chemical in your essay for the first homework, right? So the polychlorinated biphenyls are also known as the PCB, right? So for example, they have a relatively low concentration in the ocean, right? And for these phytoplanktons, actually they can take or pick up these PCB into their body, right? And these phytoplanktons can be consumed by the zooplanktons, okay? So you see that the PCB concentration started to build up in the zooplanktons because they are not easily consumable, right? These are more like the inert species that are going to build up in the body of these living species. Further, the zooplanktons are getting consumed by the smelt, and further up to the lake trout, and then to the ducks or the herring gull, right? And then you see that in their eggs, right, you're going to find a very high concentration, relatively high concentration of the PCB. So this is how the bioconcentration is getting shown up, right, in this food chain or food pyramid here. Okay, so the PCB currently is banned, right? But it has been used as a very common chemical species that are shown up in the coolants or lubricants, right? And they're being used in the, they were being used in the transformers, capacitors, and other electrical equipment because they don't burn easily. So if they don't burn easily, it means that they're very inert, and that's why their concentration can get built up the food chain, right? So the manufacture of the PCB was stopped in the United States in 1977 because there are evidence showing that they can be harmful, right? So they can harm they can lead to these harmful health effects. Okay, so we'll talk about these toxicity later on in this class too. Okay, so this is the definition of the ecosystem, right? And I think one pretty important component of the ecosystem or of this chapter is to understand the cycles of different types of elements. Okay, so there's nutrient cycles. So the first, or I would say the most important one, is the carbon cycle, because the carbon cycle also involves the life cycle of the carbon dioxide, which is a major greenhouse gas species in our planet Earth, right? So more specifically on the carbon cycle, as you can see, basically the sunlight provides energy, right? And the trees, these phototrophs, are going to go through this photosynthesis process to basically absorb or convert these carbon dioxide into organics, right? And these trees further will get decayed or they can get consumed by these animals, right? And then they can get basically deposited onto the earth, right? And for the carbon that's deposited onto the earth, a fraction of them can go to the fossil fuels, right? And on the other hand, the respiration process, for example, the bacteria or the human consumption of this meat can go through the respiration, right? We can produce energy, but at the same time produce carbon dioxide and water, and these carbon dioxide can mix back into the atmosphere. The plant themselves can also go through the respiration because overnight there's no photosynthesis anymore, so they have to consume the organics in their body. But at the same time, there are human activity, right? That's going to consume these fossil fuels and bring these carbon dioxide back into the atmosphere again. And also the ocean uptake is a pretty important section of the carbon cycle because carbon dioxide can get absorbed or can react with the species in the ocean, right? So this is a general cycle, but what is the contribution for each component here, right? So how can we quantify these processes? So here it gives a sketch of the carbon dioxide budget for the, let's say for the period of 2007 to 2016. And these numbers here are talking about the gigatons of carbon dioxide per year. So we know that the megatons, right? Mega means 10 to the 6th, right? So giga means 10 to the 9th, right? So this is 10 to the 9th tons of carbon dioxide, right? And so basically over these 10 years, each year, or the average, let's say emission or the amount of carbon dioxide getting fixed or getting absorbed are shown in here, okay? So you see that a major contribution of the carbon dioxide into our atmosphere is the fossil fuel combustion, right? Fossil fuel and industry. That's contributing to around 34.3 gigatons of carbon dioxide into our atmosphere, okay? And then you see that this is definitely putting the carbon dioxide out. What is another source? The other source is the land use change. So basically the land use change is talking about converting the forests into farmlands, okay? So if you think about the forests in the tropical region, right? So you have a lot of green trees and they, most of them do not die over a, let's say a seasonal cycle, right? It means that the carbon dioxide is actually getting stored in their body. But if you change that to farmland, and farmland, the crops will have a life cycle, right? So basically each year, although you can have the greenness from the farm, but over the autumn or during the fall or during the winter, right? they will die, right? After they're dead, they'll get decomposed, and those carbon dioxide are actually getting released into the atmosphere. So there's a net change of the carbon dioxide due to the change of the land type, right? So this is contributing around 4.9 gigatons of carbon dioxide per year. All right. So these are the major sources. What are the sinks, right? What are absorbing the carbon dioxide? So basically, the ocean is a major sink, right? So ocean takes around 8.7 gigatons of carbon dioxide, right? And also the land sink, and this is where the trees are playing a role, right? They can absorb the carbon dioxide and then basically generate more trees, right? So these are the major sinks and sources of the carbon dioxide. And if you do a simple calculation, let's say if you add up these sources, right, and then minus the sinks, minus the amount of carbon dioxide that are getting removed, and you can calculate that the atmospheric growth is around 17.3 gigatons per year, right? This is, I would say, a relatively large increase of the carbon dioxide. And this is also what is pushing, also the driver that's pushing the carbon dioxide concentration to rise up, right? I think we showed that in our first class. So right now it's hitting the mixing ratio or concentration of 420 ppm, right? It increased by almost 1/3 of the mixing ratio by or the concentration over the past 60 years. So you can see how fast this concentration increase is, right? So this is about the carbon cycle. And we also have the nitrogen cycle. So the nitrogen cycle involves fewer processes, right? Unlike the carbon dioxide, which is everywhere. So the in terms of the nitrogen, their concentration or their total amount is also lower, right? So the major processes that's involving the nitrogen is that the organics, for example, the trees or the animals or a human being, right? We contain a lot of nitrogen ourselves, mainly because our protein, our amino acids contains these nitrogens. So after these species die, right, they will have a decay process, and we're going to generate ammonium. And the ammonium can be converted into nitrites or nitrates, right? And at the same time, actually, our atmosphere is a net source of the nitrogen, mainly because the lightning process actually converts nitrogen and oxygen into nitrogen oxides. Okay, so the lightning will basically generate these NOx process, and we know that these NOx can react with water, right, to form nitric acid. So they will just go with the precipitation, go with the rainfall to get down to the ground and further participate in the cycle of the nitrogen on the ground. Okay, so this is about nitrogen. We also have the phosphorus cycle, right? The element of P, right? So phosphorus is also very important because they're being used as a part of the protein, right? In the living species, right? And for the phosphorus cycle, they're mainly happening on the ground and in the water. So the atmosphere is not participating in the cycle. So basically, most of these phosphorus are actually coming from the runoff of the rock, because we know that the phosphate, these ions can combine with different types of metals, right? They can form the components of rocks, right? So the geological or these geological processes can expose these rocks to water, and then these phosphate ions can get into the water body. And furthermore, these phosphates can get picked up by the animals or by the bacteria, and then they can get or participate into the cycles that's happening in the living species. Okay. So finally, it's the sulfur cycle. So the sulfur cycle is quite important, not only because it's associated with the let's say the living species, right? It's a part of our human body, but also the sulfur cycle is hugely influenced by the human activities. Okay, so for example, for the emission of the sulfur, it's actually mainly coming from the let's say the for example, the coal-fired power plant, right? You can see the coal-fired power plant contribute a large amount of sulfur dioxide into the atmosphere, right? And there are some other activities like the agriculture activities, right? So they can also contribute sulfur to the cycle process, right? And these sulfur dioxide, after they're in the atmosphere, they can get down through the formation of the, let's say, the acid rain, right? And also they can combine with other species like the ammonia and then form some salts, and then they can get back to our ecosystem. Okay, so the human activities that are affecting the sulfur cycle include the following. For example, the use of the sulfur containing compounds as fertilizers, and also the sulfur dioxide is being released from the power plants. And furthermore, the acid mine drainage. For example, if we're talking about mining of the metals, right? So most of these metal mines, they will contain sulfur inside. For example, the iron is combined with the sulfur, and these are called the iron sulfide, right? So basically, these can get reacted to get oxidized into sulfates, right? And further get dissolved or getting mixed into the water streams and then enter our ecosystem. Okay, so these are the four major cycles of the ecosystem, right? And then we can talk about the population dynamics. So you can see that these definitions are gone through pretty quickly. So so since they don't require a lot of engineering understandings, so we'll just try to quickly go over that. And if you have questions, you can go through the reading, and also you can talk to me. Okay, so the next one is the population dynamics. So this is mainly talking about how the population of any species, right? If they're freshly introduced into a new environment that has a limited amount of food, right? How their population is going to change. Okay, so for example, if we're introducing bacteria into a petri dish, right? And the petri dish has a lot of nutrients inside, right? The agar plate. So the bacteria, of course, at the beginning, they're not going to populate much, right? Because they want to get adjusted into the environment. And some of the bacteria species which are not suitable for the petri dish environment, they will just die off, right? So their population is not going to increase much. But later, when they realize that there is a huge amount of food, right? Although limited amount, but with a low population, there is going to be a significant increase of the population, right? So this is where we call there's going to be a log growth process. So basically, the population is going to increase so fast that in a log plot, let's say 10 to the third all the way to 10 to the sixth, there's going to be a linear, almost a linear increase of the population. So as these bacteria further consumes the food, right? And then the food becomes quite limited, then they're going to go through this stationary phase. The stationary phase is a period where you're going to have a relatively constant population, where the population that's dying equals the population that are freshly generated, right? And eventually, when the food or when the nutrition is getting consumed or getting depleted, they're going to enter this death phase where the population is going to decrease. And this is indeed happening in the in our ecosystem, or at least in the lab experiments, we can see that, right? So it happens that I have some students working with the bacteria, and they're the E. coli bacteria. Right, this is a common bacteria that's existing in our environment. And you can see that at the beginning, when you introduce these bacteria into the petri dish, there are going to be just a few colonies of them. And later on, these colonies are going to, the number is going to increase, and also the area, right? So the larger the area of these bacteria, it means that there are the higher the population. And then they're going to enter this stationary phase, right? So you're going to occupy a very large area, but you can also see that some of the areas, their color is getting darker. It means that they're decaying. They're going through this death phase process, right? And in the end, when all the nutrition are getting consumed, they cannot live there anymore. Okay, so this is how we understand the population dynamics of any species in our ecosystem. Okay, if there is a limited amount of nutrition. But right now, it seems that there's, especially, let's say, for many of the animals, right, or the, for example, the plants, right, there is almost unlimited amount of the food, right, as long as we have the sun, right? That's the major contributor to the energy on our Earth. Okay, so the next is the chapter 6 about the risk assessment. So here we will do some simple calculations or to try to understand the risk. Okay, so regarding the risk assessment, there is a common calculation to understand or to calculate, evaluate what is the risk. Okay, so one simple example is that, for example, if we know the total number of deaths, right? Let's say in 2001, there are 2.4 million of deaths because of all the reasons. It can be cancer, it can be, let's say, car crash, right? It can be, let's say, coming out of the, let's say, the with different types of diseases, right? And among these different types of or reasons of death, around 540,000 deaths were coming from cancer. And with these data, we can estimate what is the risk of dying due to cancer. So the risk of dying due to cancer, let's say in a lifetime, okay, is defined as the death number that's due to cancer, right? This is 550,000, and then divide that by the total number of deaths, okay? And you can find out that number is 0.23, which means that a person has 23% of chance of dying from cancer in a lifetime. And further, if you know what is the lifespan of a person, right, the average lifespan of the population, you can also calculate what is the annual risk. So each year, what is the risk of death due to cancer, right? So for example, we know that at that time, the average lifetime of US citizen is 75 years, right? So you can divide this 0.23 by 75 to get the annual risk of dying from cancer. That's 0.003. Okay, it sounds like a small number, but if you consider the entire lifetime, right? Around 23% is not a small number. It's almost a quarter of the death. Okay, so this is an example of cancer. So what about COVID, right? So we know that COVID also led to a large number of deaths. So here are the statistics. So by the way, you can find these statistics just from the CDC website. Okay, so if you search what is total number of deaths by COVID in 2020, 2021, what is the total number of deaths in the United States? So CDC will collect these information and make them available to everyone. So if you see here, right? So in 2020, where the COVID just had the outbreak, right? So we had a total death of around 380,000, okay? And in that year, the total death number is 3.4 million deaths. So by comparing these, you can see that in 2001, we just have 2.4 million, and now we have 3.4 million, right? So this is a structural change of the, let's say, the population group, right? So maybe it's because in 2020, there is a higher population that has a larger age, right? So that's why we have more deaths here. So if you know the total number of deaths and also know the deaths that's caused by COVID-19, you can actually calculate what is the risk, right? So if you put in a number, you can find out that this is going to be around 11%. Okay, so 11% is not a small number either, right? So we also have the data for 2021, right? You have a total deaths of 386,000 and due to COVID, right? And you also have 3.5 million deaths in total. So you also calculate the risk by COVID in 2021. I would say that these two risks are similar, right? Both around 11%. So because COVID is a let's say contributing to a large number of deaths, right? Because after all, 11% is not a small number. And that is also why, okay, 9 of the 10 leading causes of death in 2020 remain the same as 2019, right? And heart disease and cancer remain the top 2, while COVID-19 became the 3rd leading cause of death in 2020. Okay, so all of these means that the COVID-19 is indeed quite deadly. Okay, so it has a relatively high risk of leading to the death. Okay, so then if we know that, right, if we know something is risky, right, or if we don't know, want to evaluate the whether a chemical is risky or not, how do we do that? Okay, so this is an example where we can do, let's say, examine how the water quality or the water component can be risky or not. Okay, so the method we can do is we can first try to identify what are the hazards, or what are the hazards, right? So basically to determine whether the exposure to a component causes increased adverse effects for humans, and also determine what level of severity. Okay, so basically to quantify the risk of certain chemicals, right? So people can do these cellular studies or cellular tests or animal tests. So people also call these as the in vitro test or in vivo test. So these two words are Latin words, right? In vitro means doing everything in cells, right? In vivo means doing things on animals, right? So basically you can expose certain chemicals to cells and see how much or how fast the cells are going to die, right? And you could also use live animals, for example, use rats or use, let's say, rabbit, right, to do these lab studies. So right now there's a stronger push to conduct more studies to the cells because of the humanity issues, right? We don't want to kill too many animals just to test or to see whether a chemical is toxic or not. Right, but from these studies, people can establish this dose-response evaluation or dose-bounce relationship. So basically, that's the relationship between the dose, right? That is the milligram of the chemical per kilogram of the body weight, right? And the incidence of the adverse reaction. Okay, so generally, these dose response curves are going to follow the Gaussian distribution. So what it means is that, let's say we're trying to expose certain groups of cells by a chemical, let's say the mass of mercury. Okay, so at the same time, we can, on the y-axis, we can plot, for example, the number of cells become dead. Okay, so as you increase the mass of the mercury, right, at the beginning, the number of dead cells is 0, right? And then as the concentration further increases, you're going to have a higher death, right? And then at a certain point, you're going to have a peak. And then furthermore, since all of the cells or most of the cells were dead, right? And then the cells becomes dead are going to get lower and lower, and eventually all of the cells become dead. And if you integrate this curve, okay, so if you convert this curve into a cumulative fashion, or the percent getting dead, okay, so then you're going to have a curve that looks like this. So basically, at very low mass of the mercury, you're not going to have any cells that are dead, right? And at very high mass, you're going to have almost 100% of the cells being dead, right? And this is where people also could define the value of LD50, right? LD50, the full name for that is the lethal dose for 50% of the test animals or test cells, okay? So basically, this is the dose where you have 50% of either the cells or or the animals became dead. Okay, so people use the LD50 to determine what is the amount or what is the dose that becomes quite lethal. So now if I ask you the question, if a chemical has a small LD50, let's say chemical A has a small LD50 of 1, let's say 1 milligram per kilogram, and B has an LD50 of 10 milligram per kilogram, which one is more lethal? It's going to be A, right? So because if you just put in 1 milligram per kilogram, it's more toxic, right? It's already killing 50% of the population, right? So this is about the risk assessment and the definition or the process where we define or we find out whether a chemical is toxic or not, or is lethal or not, right? And then finally, I want to talk a little bit about the research in my lab. So as you know, my research is mainly about air quality, right? The air pollutants. So one major reason why people are so interested in the air quality is that we know these particles, once they're getting inhaled, they're going to cause some health effects, right? And especially if in recent years, there is I would say, a surge of users or people smoking e-cigarettes. Okay, I don't know if any of you are using e-cigarettes. If you are, please try to quit as quickly as possible. Okay, so right now, based on the study in the United States or survey in the United States, actually there are more than 2 million US youth currently using the e-cigarettes. Okay, for example, in terms of high school students, there's more than 11% of all the high school students actually using the e-cigarettes. And even for the middle school students, there's close to 3% of them using these e-cigarettes. So these e-cigarettes were originally designed as a transition cigarette or transition product for the tobacco cigarettes to quit smoking. Okay, so if a person that uses a lot of tobacco cigarettes, doctors actually, or the advertisement actually try to brand these e-cigarettes as a product where, well, you can temporarily use the e-cigarettes, try to phase out, and then eventually you will quit smoking, right? Because these e-cigarettes can contain nicotines inside. But later on, these advertisements becomes more targeting at the younger generation, because smoking or vaping can generate a lot of smoke, right? And then at the same time, you can put a lot of flavors inside. For example, you can put menthol, you can put, let's say, apple, banana, different types of flavors. And it becomes, I would say, quite fashionable among the younger generation, right? And it's causing some problems. For example, right now, apart from these population or these numbers here, actually among the youth who are currently using the e-cigarettes, about 2 in 5 actually use the e-cigarettes frequently, and about 1 in 4 use e-cigarettes daily. Okay, and this disturbingly high risk or high rates of these e-cigarette usage also suggests that there might be a dependence on the nicotine, right? So basically, the younger generations are getting more and more dependent on these chemicals. Okay, and currently around 85% of these e-cigarettes are flavored. So basically, there are additives getting mixed into the liquid that's in the e-cigarettes. And I'm not sure if any of you followed the news during the COVID, actually. Actually, it's right before the COVID, we had an outbreak of disease that's associated with the e-cigarette usage. And that was called the e-cigarette or vaping product use associated lung injury. So people also call that as EVALI, right? So at that time, I think it was also in the breaking news that many young people were actually getting hospitalized because they were using a special type of e-cigarette. Actually, they add a special component, that is the vitamin E, right, into the e-cigarette, because adding vitamin E into the liquid can actually generate more smoke and makes the feeling of the smoke at the throat area very different. Okay, So many people want to try that, and eventually it appears that vitamin E is healthy or is okay when we're digesting them through our mouth. But once they're getting generated into the smoke, it's actually a toxic species, okay? Once they get into our lung, it's toxic. So for that EVALI event, close to 3,000 people got hospitalized, right? And then the total death number is 68. And remember, right after this event, there was the outbreak of COVID-19. So basically, the study on these e-cigarettes aerosols were kinds of stagnant or I would say influenced a little bit because of our rapid response to the COVID-19 disease transmission. So what we did in our lab is that we want to estimate the risk of e-cigarette aerosols, and then to study, well, the health effects, right, of these tiny particles. Because anyway, these e-cigarette actually are generating these aerosols or particles, and they're getting deposited into human lung, right? So in this study, actually, we did not use human subjects or animals. So we were just using the human cells. Actually, it's the one type of the standardized cancer cells. Okay, so cancer cells can grow by themselves, right? So we were trying to determine I would say a value that or what is toxicological effect or toxicity of these aerosols, kind of trying to understand the LD50 of these aerosols. Okay, so here I also have a short video that's showing how we introduce the e-cigarette aerosols into a chamber. So you can see that here we have the petri dish inside. Actually, we culture the cells inside, and then at the same time, this is I would say a popular e-cigarette. This is called the Wupu e-cigarette, right? And we actually generate these aerosols and then deposit them onto the cells and see how the aerosols, how the particles are killing the human cells. Okay, so let me quickly share this video. So basically, we're using a vacuum pump, right, to pull the aerosols or pull the airflow through this e-cigarette. And then at the same time, for this e-cigarette, it has an on and off switch, right? So if you press that button, you're going to generate these aerosols and they're being introduced into the chamber. So how does it look like? What is the result, right? So let me go back to the slides again. Okay, so we basically determined what is the cell viability after we expose these cells with 30 puffs of the e-cigarette smoke. Okay, so as a reference, so for a general e-cigarette smoker, they're going to use around 168 puffs per day. Okay, so we first determined the cell viability. So basically what we did was we first cultured the cells onto these petri dishes, right? And we count what is the number of the cells, right? We also have control groups that are not exposed to these e-cigarette aerosols. And then we expose these cells with different types of the aerosols. For example, for that e-cigarette, you can actually change the power that's being used on the e-cigarette. The stronger the power, the more aerosols you can generate. Okay, so you can see we tried 10 watt, 35 watt, and 60 watt. Okay, and also we changed the percentage of the nicotine that's in the solution. Okay, there can be 0%, there can be 6%. So you may wonder why there's 0% nicotine for the e-cigarette smoke, right? So basically, this is where the e-cigarettes are attracting the younger generation, because even if they don't need nicotine, they can still use that for fun, right? They can generate a lot of smoke, and that looks really cool, right? It's basically it has been very trendy in the social media, and this is not a good thing to their health. Okay, so for example, if you see here, right, if we expose, even if we just use a power of 10 watt, you see the cells actually has around 25% decrease in the viability. Okay, if you increase the power all the way to 60 watt, the viability reduced down to below 50%, which means that they are indeed toxic, right? They're leading to the cell death. So you may say, well, is that really toxic, right? 30 puffs. So basically here, one thing we also need to do is that we need to interpolate the dose to the actual human body, right? Because right now, we're just counting, or we're just depositing these aerosols onto a small area of the cells, right? So in terms of this area, it might be just, let's say 20 centimeters squared, right? And if you compare that to the entire area of a human lung, right, that is what, close to 1 meter squared, right? 1 meter squared is much, much larger than 20 centimeters squared. So which means that basically we're actually depositing a large amount of these aerosols onto the cells, right? So we need to further do a dose interpolation to see how the realistic deposition or realistic dose of the e-cigarette aerosols can kill the human cells. Okay, so I also conducted some studies regarding the how the primary aerosols and secondhand aerosols, these e-cigarette aerosols, might affect the cell viability, because I think one advantage, I would say one characteristic of the e-cigarette is that unlike the tobacco cigarette, where once you light it up, you always generate smoke. For the e-cigarette, there's a switch, or it's activated by inhalation, right? If you're not using that, you're actually not generating any secondhand smoke. But the thing is that for a e-cigarette user, when they exhale, they're still going to basically push these aerosols out, right? Because not all the particles get deposited into the lung. So we actually generated some of these simulated secondhand aerosols and expose them into the, or introduce them into the chamber and expose them to the cells. So you can see that for the secondhand aerosols, indeed, they can have a higher viability. Okay, if you compare this one and this one, right? The blue one represents the secondhand smoke, right? The blue one is always a little bit higher than the red one, but the secondhand aerosol can still reduce the viability, right? You see, it's at least not 100%, which means that actually, even if you're not using the e-cigarette, and if you have friends or you have families using e-cigarettes, their exhaled aerosols can still be quite toxic to you, right? So cell viability can be one factor, one indicator of how toxic it is, right? There are also other factors. For example, people conducted these what we call the tear measurements. So this is basically measuring the membrane electrical resistance. So let's say these are the cells in our tested setup, right? What we could do is that we can measure the resistance at 2 different locations of these cells, right? And then in general, if the cells are still alive, then their membrane is going to have a relatively high resistance, so they cannot conduct the electricity really well. But once they become dead, right, the resistance becomes lower. Okay, so you're going to have a lower tear value. So basically, they can conduct more electricity. Okay, so we conducted these measurements onto the cells too, right? You can see for the control group, if we were not exposing the e-cigarettes to them, the cells were still growing, right? So their tear measurement or value is actually increasing. It means that they're living pretty well. But once you expose them to the e-cigarette aerosols, either 10 watt or 60 watt, you can see that their values becomes lower. It means that the cell membranes are getting damaged, right? And some of these cells became dead. Okay, so these are different indicators where people can conduct these toxicity measurements. So as I said, these are the ongoing projects in my group. And if any of you wants to know more, you can feel free to reach out to me. Okay, so I think this is also the end of this class. Okay, so as you can see, this class has quite many definitions. Okay, so not so much on the engineering understanding or not so much on the calculation. So next class, we're going to talk about the hydrology. And if you have any questions, just please feel free to let me know. All right, see you next week.